DNA structure. Nucleic acids. Nucleic acids are large molecules, polymers, consisting of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They store genetic information, it's one role, and instruct the cell in creating proteins. There are two main types of nucleic acids, DNA, and in some cases, RNA is the primary source of heritable information. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. DNA will be discussed in this topic. Nucleotides. Nucleic acids are composed of chains of nucleotides. So the nucleotide is the monomer. When you put a bunch of those nucleotides together, you build up to the polymer that is the nucleic acid. Nucleotides have three main parts: a sugar, here, a nitrogenous base, also just called a base, and a phosphate group. Nucleotide sugars. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. You can see them side by side here. Here's the difference. On this carbon, ribose has an oxygen, and deoxyribose does not. Nitrogenous bases. DNA contains the bases adenine, guanine, cytosine, and thymine. So A, G, C, T. RNA contains the bases adenine, guanine, cytosine, A, G, C, and uracil, U. The difference is that DNA uses thymine rather than uracil. Nitrogenous bases composed of two ringed structures are known as purines. So adenine and guanine have two rings. These are known as purines. Nitrogenous bases composed of one ringed structures are known as pyrimidines. Here's one ring: cytosine, thymine, and uracil. DNA is a nucleic acid made of two chains of nucleotides. In each chain, the nucleotides covalently bond together, forming a strong sugar-phosphate backbone. So you can see the sugar covalently bonding to the phosphate group. The bonds connect the sugar on one with the phosphate on the adjacent nucleotide, illustrated by the gray pentagons and blue circles. So the sugar from one nucleotide covalently bonding with the phosphate of an adjacent nucleotide. That would be this one, and also this one bonding adjacently here. The two chains are connected with hydrogen bonds between the nitrogenous bases. So between the cytosine, guanine, adenine, thymine, you see these lines. These are the hydrogen bonds. The hydrogen bonds are much weaker than the covalent bonds that hold the sugar-phosphate backbone together, making it easier for complementary strands to separate. So we can separate these two strands because we can break these hydrogen bonds to allow the DNA to replicate and be transcribed into proteins. DNA functions. DNA stores genetic information that is used for structure, daily functions, and for heredity. Genes are made of DNA and encode all the molecules required by an organism. DNA is the main component of chromosomes. Chromosomes are copied and passed on from parents to offspring during reproduction. Eukaryotic and prokaryotic chromosome structure. The structure of chromosomes is different in prokaryotes and eukaryotes. Functional differences will be covered in the transcription and translation units later in the course. Prokaryotic chromosomes are found in the cytoplasm and are circular in shape. Plasmids are small strands of DNA which are found in the cytoplasm of bacteria and protozoa and replicate independently of the nuclear DNA. They are used in genetic engineering. Eukaryotic chromosomes are found in the nucleus and are double helixes. There are multiple linear chromosomes. A unique set of chromosomes are found in mitochondria and chloroplasts. Some eukaryotic cells have plasmids. So here are some differences between prokaryotic and eukaryotic DNA. In prokaryotic DNA, we have bacterial chromosomal DNA here, and then the circular plasmid DNA, both in the cytoplasm. Eukaryotic DNA exists as this double helix inside of the nucleus of the cell. Let's take a look at the nucleic acids present in DNA. The 4 types of nitrogenous bases are illustrated in red, purple, yellow, and green. The bases are abbreviated as adenine A, thymine T, guanine G, cytosine C. The bases in one chain, maybe the one on the right, bond with the bases in the other chain on the left with weak hydrogen bonds that you can see indicated by the lines in between. The bonds between nucleotides in each chain are called phosphodiester bonds. Like bonds between monosaccharides, they are formed by dehydration synthesis reactions. So here's one nucleotide, G. We have the nitrogenous base G. So if I just put a box here, right? And then here's a neighboring one, T here. You can see the formation of this phosphodiester bond between the phosphate of one nucleotide and the sugar of an adjacent nucleotide. Let's talk about the prime notation. You see here this says 5' end, 3' end, and this says 3' to 5' phosphodiester bond. So the prime notation indicates the number of the bond formed by the sugar's carbon atoms. It starts from the bond with the nitrogenous base, ending in a clockwise direction with the phosphate group. So do you see how I start at the carbon with the nitrogenous base as 1'? Same thing here, 1'. And I go clockwise. So this is 1', 2', 3', 4', 5'. Same thing here, 1', 2', 3', 4', 5'. So what happens is the 3' carbon of one sugar bonds to the 5' carbon of the adjacent sugar through the phosphate group. The bonds are key to the operation of DNA. The strong phosphodiester bonds create a strong molecule. The weak hydrogen bonds allow the DNA molecule to be split, copied, and then put back together, critical to the duplication of the DNA molecule. Complementary bases. The base pairs that bond with each other are called complementary bases. In DNA molecules, hydrogen bonds between complementary bases in the rungs of the ladder hold the backbones together and at a constant distance apart. So we can see these hydrogen bonds, 2 of them forming between thymine and adenine, and 3 of them forming between guanine and cytosine. A purine only bonds with a pyrimidine because of their structure. This ensures that the diameter of the DNA molecule stays constant as each rung includes 3 rings. Remember that purines are 2-ring structures, pyrimidines are 1-ring structures. So if a purine and a pyrimidine bond together, we always end up with a 3-ringed rung. A further restriction is that adenine only bonds with thymine and guanine only bonds with cytosine. So you can see throughout this image, always adenine with thymine, AT, guanine always with cytosine, CG. Cytosine and guanine form 3 hydrogen bonds, while adenine and thymine form 2 hydrogen bonds. This creates the double-stranded helix because of the angles of the strong sugar phosphate bonds. DNA is anti-parallel. Each strand has 2 ends, a 5' end and a 3' end. The 2 strands of DNA always run in opposite directions. They are anti-parallel to each other. The 5' end has a phosphate group at the end. You can see that here and here. The 3' end has an OH group at the end, here and here. The 3' position where this OH group is the place where the next nucleotide is added to the growing strand. So we'll create another phosphodiester bond at this 3' position in order to keep the DNA strand growing. Each strand is unique due to its sequence of bases. In this way, genetic information is stored in the sequence of nucleotides. Since the nitrogenous bases T, C, A, and G are not part of the phosphodiester bonds, the base sequence is independent of the backbone, and therefore any base sequence is possible.